Hello, my name is Benjamin Pepper, and I am a junior chemical engineering undergraduate at the University of Massachusetts Amherst. I'm a part of Dr. Shelley Payton's Soft Materials Biomaterials Lab, and I am here today to present about visualizing astrocyte response to mild traumatic brain injury.
Okay, so I'm just going to start off by just giving a little introduction to minor traumatic brain injury. And TBI is what traumatic brain injury stands for. It's pretty much just like when you hit your head hard, when you get a concussion, that's what traumatic brain injury is. And there's three different ways it can happen. It can happen through direct impact injury, as you can see in this image. So pretty much indirect impact injury, what happens is that you hit your head hard against the surface, and that's just like all concentrated right in a small area. And then over here we have the second type of traumatic brain injury, which is acceleration deceleration injury. And this is really like whiplash, like if you're in a car crash and your head goes forward and slides backwards, that'll be that different whipping motion, and you'll see all of the injury is absorbed here and here because that's where the brain motion is. And then the third type of injury is a blast injury, when like a shock wave gets sent just through the body and through the brain, and it creates these cavitation bubbles. And in all of these situations, you can see right here, and right here, there's all these little red dots. And these red dots are cavitation bubbles. And pretty much what that means is in traumatic brain injury, a cavitation bubble will build up because of the trauma that the brain has experienced, and they'll eventually bubble up enough that they'll burst, and this will send a microscopic shockwave through the brain, which isn't good for it. And what is different and important and special about minor traumatic brain injury is that it's difficult to detect and diagnose. So with normal concussions, you can take tests and really see, did this injury I sustained, did this give me a concussion? But with minor traumatic brain injury, it's that level right below where you're really able to detect it. So right now in just industry and just in life, it's just hard to detect and diagnose minor TBI. So the main cells that I'm really looking into to see how the brain responds to this mild TBI is through astrocytes. And astrocytes are just nervous system cells that transition from a quiescent state to a reactive state in response to injury. So pretty much they start out as just these normal, they have this body and they have a bunch of just branches in which are called projections, all of these different branches that just allow them to communicate with other cells and really allow for the brain to have good protection from the blood, because what the astrocytes are really responsible for is the blood-brain barrier. They make sure that nothing really bad and harmful can go into your brain so that it doesn't get as many infections or isn't really as affected by harmful things as the rest of the body is. But when you experience these cavitation bubbles that you see in traumatic brain injury, what happens is that the shock waves affect the astrocytes in a way that it turns them to this reactive state, which causes them to express and secrete different proteins. And what we really want to see is we want to investigate, how that just traumatic brain injury, how it causes this transition to happen. We want to see if we're able to see these morphological changes and really see these proteins that are expressed and secreted. So as I kind of alluded to before, the real main research question that I'm looking into is the question of, can we elucidate the extent of a brain injury and observe the acute response of astrocytes to mild TBI cases that would otherwise be undetectable in brain tissue? Really, we're trying to fill in this hole of just an inability to really see and detect brain injury when it's there, so that people know that it's there, know to be careful, and know to not do stuff like that in the future, so that it's not a compounded thing that just becomes worse and worse for a person. So our first big research question when we were looking into this was, how are we going to be able to model traumatic brain injury? How are we going to be able to actually make sure that this is a thing, and how are we going to be able to see that what we want is actually there? So we did this through the use of needle-induced cavitation to simulate cavitation injury. And as you can see here, this is the whole apparatus of needle-induced cavitation. And pretty much what it does is it moves this plunger very slowly that has this liquid in the syringe that is read by a pressure sensor that has a needle that will go into whatever we have as our brain, and it will blow a bubble into this material, and the bubble will expand, and it'll collapse, which will end up sending a microscopic shockwave through whatever material is down here, and that will sort of simulate what happens when the cavitation bubbles burst in mild TBI. This is what it looked like in reality. You have your plunger is off screen. You can't see it over here in the syringe. And this is just tubing that's able to connect the syringe right to the needle. And then this is our material that we're blowing our bubbles into to model traumatic brain injury. Our next big challenge we experienced was great. Now we have a way to model mild traumatic brain injury. But how are we going to be able to model the brain? Because we can't just take a whole brain and just throw it on there, because we won't be able to see anything under the microscope. That's not really feasible. There's a lot of other stuff in the way. So what we came up with was using biomaterials, was using collagen hydrogels to model the brain. So what we came up with was using a 10 microliter collagen hydrogel network with astrocytes dispersed throughout. Astrocytes were grown in culture, and in the making of the collagen hydrogel, astrocytes were inserted in at a concentration of 10,000 cells per gel with a 4 milligram per milliliter collagen concentration. And this was done in order to obtain a literature-seen desired elastic modulus of the brain. Because what we want in our material is that, first off, we want the astrocytes to be in there. We really want just to see that everything is working out. But we also want to mimic that kind of texture and kind of just jiggliness that the brain has, because that's super important in making sure that anything we do is properly replicated. So in doing this, we were really able to get that feel, and as you can see here, this collagen hydrogel is very, very small, so we're able to make a lot of them. We're able to really fine-tune them and see exactly what we want to see, and able to grow astrocytes in and make it work. So these are images that were taken during the needle-induced cavitation process that was done on collagen hydrogels that had astrocytes. As you can see in these images, that the collagen hydrogels are really, really small, especially compared to just the whole apparatus. So right here, this is just pre-needle insertion. You start by just making sure that the needle is right above the collagen hydrogel, just to make sure that when it goes down, it will actually go down into the gel. Then the needle is inserted into the gel, and it's made sure to not puncture the very bottom, just so that the needle is right in the middle and that you're able to blow bubbles in and really get that cavitation going. And then this is what it looks like, the gel post-NIC after the recording had been stopped. So how it works is that there's the whole pressure sensor that's connected to it. That's the important thing, to make sure that we're properly mimicking the brain, because that's definitely something that we wanted to do. So we started the run, then we started the syringe so that this liquid started to be pumped out, and then it started to bubble into the gel, and then once it got to this point where we could clearly see the liquid coming out, this red dye, it's pretty much just distilled water and red fruit dye, and we wanted to use this to really see the spread and to see how it went through. But once we saw that it was leaking out, we'd stop the run, we'd stop the pressure sensor, and go from there. So these were some of the resulting graphs that we got from the NIC runs. And as you can see here, this is when we just started the run and nothing was going on. It was just constant. And then when we started the inflow of the syringe, that's when we started to get this big pressure jump. And then it started to crash back down again, right where the material couldn't handle it anymore, and that's when it bursted. And right here at this maximum pressure, we were looking at to see what our elastic modulus was, which we wanted to be around 1 kPa, which is right around where the brain is. And for values we got here, we got 0.915 kPa and 0.918 kPa. So it's definitely very close to where we want it to be, but we're going to try and just slightly increase the concentration of collagen, really fine-tune, make sure that there are exactly 10,000 cells in each gel, to make sure that we're actually getting exactly what we're saying is in there, and making sure that we have that modulus that is very similar to the brain. So these are some images of the astrocytes taken in the gels on the first day that they were treated, so day 0. As you can see here, all of these ones that aren't in focus, these are cells that are in different planes. So the really nice thing about using a hydrogel as opposed to just using a plate is that we're really able to get cells growing everywhere. It's able to mimic the network and communication that astrocytes will have in the brain. Our collagen hydrogels we like to think of as little mini brains. So as you can see here, this is an astrocyte that has a lot of different projections. One of them was measured using image J of 20.05 microns, and this is in 20x. And as you can see here, there's a bunch of astrocytes. We were able to get astrocytes growing all throughout the gel, looking pretty healthy and looking pretty good. So needle-induced cavitation was done on the gels 2 days after the cells were seeded to give the cells a bit of time to just get used to being in the gels and make sure that they were comfortable and able to grow. As you can see from these day 2 pre-NIC images, there's a lot of projection growth over here. And these are 2 astrocytes from the same Z-stack, which pretty much just means that these are 2 astrocytes on different layers and different levels inside the gels. There's a lot of healthy growth going on here, and we could see that these cells were definitely comfortable with the gels and ready to experience the NIC. So these are images taken on day 4 after the astrocytes were placed in the gels. So this is taken 2 days after the NIC occurred. So when the NIC occurred, the gels were taken out of the place that they were adhered to. So they did the cavitation on them and then put them back in. So they were kind of just floating around in solution, which made it really hard to take pictures of. But these are the pictures that we're able to get. We can see here that there are still some projections, but it really doesn't look as good as it did before. So definitely for the future, we're going to make sure that doing cavitation right into the wells so that they can stay adhered, so that we can have consistent photos to have throughout is definitely going to be a must. But these are promising starts that the cells are still there after the cavitation occurs. So the conclusions that we can draw from this is that the cells definitely do look different pre and post cavitation. So we can see that the cavitation definitely impacts astrocyte cells. And we can see that astrocytes are affected by the fact that this cavitation and mild traumatic brain injury has occurred. Going forward, we're going to look into working on performing a live dead assay to determine cell death as a function of distance from entry location to see really how the cells in different locations respond to the cavitation. And we really want to perform the cavitation under the microscope so we can get a recording and see a live view of how cells respond. This is really promising stuff for the future that I'm super excited to look more into next year. Thank you all for taking the time to listen to this presentation. I want to give a special thank you to Dr. Shelley Payton and Carrie Dugan for helping me along with this research.